Looking at the actual functioning unit of a muscle and how it contracts, we're going to expand a little bit on what we just looked at in terms of the structure of a muscle. And here we have a quick review of that. We're looking at the brachioradialis muscle here in the forearm. So first off, we have the full muscle, which is represented by the red circle, and that's the muscle belly, the full cross-section of the muscle tissue itself. The muscle belly is comprised of a series of fascicles. So these are the smaller groupings of muscle tissue inside the muscle belly. And then we learned that each fascicle is made up from a whole series of muscle fibers. Now that's as far as we went with the first initial lesson on the structure of muscle. But to continue this lesson, we're going to look at each individual muscle fiber and understand that it's made up from a series of myofibrils. So the myofibril itself is the smallest strand of muscle tissue, and it's made up from a consecutive series of structures called sarcomeres. Now, the sarcomere is what I like to refer to as the functioning unit within muscle tissue. It is this structure right here, and we're going to learn all the different components of a sarcomere, that actually allows the muscle to change length and pull on the bones. And that allows us then to create movement by changing the angles of the joints. So what is the sarcomere comprised of? Well, the sarcomere is made up of a number of different number of different structures, and we're going to start by looking at the definition of the end point of the sarcomere, and it is what's referred to as the Z line. So when we're looking at any structure or any schematic image of a sarcomere, it's going to be defined by one Z line at one end and another Z line at the other end. So that is the edges of the functioning sarcomere. Now, within those Z lines, we have two different types of filaments. The first one, shown by this schematic here, is referred to as the thin actin filament. And we have thin actin filaments on each side, almost looking like combs pointing in towards the center. Now, inside the thin actin filaments, we have these bigger, thicker filaments called thick myosin filaments. So we have thin actin and thick myosin, and they interlock with each other, as you see here, with the thick myosin filling in the space between the thin actin. And if you put both of your hands together and interlock your fingers and slide them back and forth, you're doing a really good job of demonstrating how the actual contractile process works. So what actually happens here is the thick myosin filaments have these little structures on them called myosin cross bridges. And what happens during the whole process, these cross bridges connect the thick myosin to the thin actin, and under certain circumstances they fold and they pull the ends of each sarcomere closer together, and they fill up this space called the H zone, where this side will slide in and this side will slide in, decreasing the space. And if every single one of these sarcomeres does this same thing within a myofibril and within a muscle filament, within a fascicle, and within a muscle belly, the end points of that muscle come closer together, creating movement. So the labels that I want you to understand when we're looking at the sarcomere are the sarcomere itself, which is the functioning unit of the muscle. It's defined by two end Z lines. Within those Z lines, we have two different types of filaments, thin actin on left and right, and we have thick myosin in the center. The thick myosin fills up an area of space called the H zone, which is the space that will decrease when contractile function occurs. And that occurs by these myosin cross bridges folding and pulling in on the thin actin filaments. So let's take a look at this step by step on how an actual muscle contracts. But before I do that, let me just jump over and show you a little animated GIF, which is going to demonstrate this. There we go. So here we have our thin actin filaments, and we have our thick myosin filaments. And you'll notice the myosin cross bridges that are attached to the thick myosin filaments will fold. And when they fold, they pull the thin actin filament in, decreasing the H zone, and overall decreasing the linear length of each muscle. And that's how an actual contraction occurs. So, step by step, how does that actually happen? Well, again, the functioning muscle unit is the sarcomere, and it contracts through the following sequence of events. Number one, an electrical impulse is sent from the brain along the nerves to the muscle tissue. This electrical impulse triggers the release of calcium within the muscle cells. And it's this calcium that triggers the whole system. So the presence of calcium activates the myosin cross bridges to fold. And when these structures fold, they pull on the thin actin filaments. The myosin filaments decrease the H zone, so this space in the middle decreases because both sides are pulling in. This sliding motion decreases the overall length of the muscle by shortening the length of individual sarcomeres. And when all these sarcomeres, millions and millions of them connected together, start to shorten, the result is an overall shortening of the muscle length, which creates a pull on the tendon and bone attached to create movement within your body. And that is all on page 14 of your coloring book. So again, there we have our muscle structure all the way down to the functioning unit called the sarcomere. There we have some basic anatomy, a schematic of the anatomical structure of the sarcomere. And here we have step by step the process which allows a muscle to contract. And then again over on my there we have a animated GIF showing the process in action.